So, good morning. For those of you who I don't know, I'm Denise Jamison. I'm the Vice President for Medical Affairs and the Dean of the Carver College of Medicine. We are so proud to be hosting the fourth symposium and introducing our newest cohort of scholars, Stead Scholars, today. Um, it's going to be really exciting this morning to learn more about the work of our three early career faculty members who are making advances in their respective fields of research. So, on behalf of the entire Carver College of Medicine, I'd like to extend a special welcome and thank you to Jerre Stead, along with his wife, Mary Joy, who's made this program possible since 2022. Mary Joy is here in Iowa City, but not able to join us this morning. But we're recording the event so that she and others can watch and learn more about the work of the 2025 cohort of Stead Scholars. We are so grateful to the generous and ongoing support from the Stead family that makes this transformational program possible. We are also pleased to welcome Jerre's colleagues, Tiffany Shaw and Dana Larson from Stead Impact Ventures, which focuses on impacting healthcare globally through its innovators network and mission-aligned companies. And the three of them are all sitting in the front row. We are, again, so grateful for your support in helping today's faculty presenters pursue new ideas in biomedical research, new ideas that have the potential to become meaningful discoveries in human health and disease. So I'd also like to spend a few minutes introducing our newest cohort of Stead Family Scholars, who you'll be hearing more from in just a few minutes. So when I call your name, if you would please stand and remain standing to be recognized. Dr. Stephanie Gantz, Dr. Jeremiah Johnson, Dr. Linda Sun, and Dr. James Byrne, who unfortunately could not be here today due to a family emergency, but he did ask us to share his gratitude for this honor and express that he looks forward to sharing more about his research with us at a future opportunity. So let's give our new Stead Scholars a round of applause. Thank you. Although we're introducing and celebrating our 2025 cohort today, I'd also like to recognize the Stead Scholars from our three previous cohorts. So again, if you would please stand and remain standing when I call your name. Dr. Georgina Aldrich, Dr. Denise Adesoy, Dr. Rebecca Dodd, Dr. Lindsay Harshman, who couldn't be with us today, Dr. Alejandro Pazula, Dr. Amy Ryan, Dr. Jennifer Burmick, Dr. Manir Tunas, Dr. Mary Weber, and Dr. Ling Yang. Let's recognize these scholars as well. Thank you. So these individuals are making great strides in their respective fields, and many of them have built their research programs from the ground up. It's inspiring, and they all deserve major kudos. Since we're limited on time, I'm not going to go specifically into the work that these scholars are pursuing, former or current scholars, but I encourage you to check out more of their research on our Carver College of Medicine website. So before we hear from our current scholars, I'd like to share some background information. The Stead Scholars Program was established in 2022 to recognize and support the development of outstanding early career scientists. And we think about early career as tenure-track faculty in our college who currently serve as assistant professor or who have become a tenured associate professor within the last 2 years. In addition to being early in their careers, the faculty members selected for this program are on their way to becoming nationally and internationally recognized leaders in their areas of research. Each of these investigators has built an impressive record of achievement and productivity, demonstrated in part through federal extramural grant support. Impactful published research, along with evidence of innovation and collaboration in their work and a growing reputation in their field of research, also are criteria used to determine and select the investigators for this program. To put it simply, The Stead Family Scholars are some of the Carver College of Medicine's brightest stars, individuals whose work will contribute to and perhaps even reshape the body of scientific knowledge and medical literature. This cohort each receives $125,000 per year for 3 years to support their research efforts. They also are provided opportunities for communication and leadership training, which will strengthen their ability to articulate the importance of their science in a compelling and accessible way, prepare them to lead the next generation of scientists, and help accelerate their careers. And we had a great session before this brainstorming about what additional things could be provided to both the current Stead scholars as well as the cohort of scholars who have completed the program. The program supports faculty from across the college. Some are in basic science, others are doing translational research that overlaps with their patient care practice. So, as I've noted, this program would not have been a reality without the support of Jerre and Mary Joy, who are originally from Moki Makokata. I knew that, and I've said it correctly before, but at the stress of the moment, I screwed it up. So apologies. I like to say that I'm a new Iowan, 2 years now, but I'm still learning. Jerre earned his Bachelor of Business Administration degree at the University of Iowa in 1965 and has enjoyed a long and successful career in technology and information. Mary Joy is a member of the UI Center for Advancement Board of Directors, an active member of the board's Development and Alumni Committee. And together, the Stead served as honorary campaign co-chairs for the university's For Iowa Forevermore campaign from 2006 to 2016, which raised an incredible $834 million for our health system. And during that fundraising campaign, Jerre and Mary Joy made their own transformational gift commitment for research, clinical care, faculty development, and of course, our number one ranked children's hospital in Iowa. In recognition of their dedication and their generosity, the university named the Stead Family Department of Pediatrics and UI Healthcare Stead Family Children's Hospital in their honor. So, Jerre, so many thanks to you. Thanks for your friendship. Thanks for your commitment. It's now my pleasure to welcome Jerre to the stage with a very special announcement to share.
It's great to be here today, and I was actually in the hospital a year ago at this time, so I missed last year, so I'm catching up. And I must tell you, it's just such a pleasure to see the scholars, to have the time like Denise just said this morning. We learned a lot, like we hoped to by listening, things we're going to do in the future. I just want to give you 2 minutes of how this all came to be. Mary Joy and I, as Denise said, lived here. We lived in a 40 by 10-foot trailer for 4 years, lived on $2,900 a year. I worked full-time, 40 hours a week, plus carried 17, 18 hours. We had both of our children here in this hospital. And I can still remember way back when that happened, when Jay, our number 2 son, was born in December of 1963. And it was in the middle of, it was midterms for me. And so I was up all night because Jay was delivered just fine, but Mary Joy had a few problems. And so I go into my professor that day and I said, could I take the test tomorrow? Because I'm really tired. And he said, you're a great student. Don't worry about it. Take the test. So I got a C. And so I had straight A's, and those meant a lot to me at that time. So at the end of the semester, I get a B+. So I go back to the professor and I said, do you remember when you gave me the C because you made me stay up all night? He said, yep, I remember it. And I said, that B+ is not very happy for me. And he said, I'll never forget. He said, what's the difference between an A- and a B+? And I said, nothing probably, except in my career is going to be crippled for the rest of my life, which it wasn't, of course, but I'll never forget it. But that all happened in this hospital. And Mary Joy and I always felt that when we had the opportunity, and we've been gifted so much over the years to be able to give back uh, and to enjoy the joys of giving, of time, talent, and treasure. And we care and love this university, this state, and we're so excited. Of all the programs we've done, this one, the Stead Innovation Scholars, which is what we are now shifting to, and you'll see that family disappear, even though the family's with it forever, and it will now say Stead Innovation Scholars. And we're going to take it the next steps. We've got a lot of great ideas that we'll communicate in the months ahead, but how do we take this wonderful talent that you all bring and take it to the next steps? The SIV, the Stead Innovation Ventures that Tiffany and Dina are running for me and leading in a great way, has 10 startups, and we'll continue to add startups. My hope is that some of the Stead scholars end up with some of those startups. And what we'll do is try our best to make this a role model of what the University of Iowa can offer that nobody else can. And we'll tie it to the business college and other places so that we're able to demonstrate why what we all know is true, which Iowa is truly the best place to be and always will be, and how we'll continue to attract even great people for many years to come. So you'll hear a lot more about that, but it's fun to be able to announce that the innovation scholars that we have, and I think are the world's best as they exist today. And I'm so eager to hear from them this morning. One of the things that Denise mentioned that's so true, when we set this up, we asked that we make sure that we provide training for each of the award winners in presenting. And one of them was talking this morning about how easy or how tough did you think it was for your elevator speech? How tough was it?
I learned that my usual one was a worthless. Well, once I got a great feedback, that was also thanks to the event.
And now you've got it cold, right? And that's part of the life we want to have is uh, I think the best way to learn is to teach and share and listen. And that's what we're going to do for the rest of the morning with really outstanding, wonderful people that are doing great work. So it's a great pleasure to turn it over to the dean, I believe.
So I have the fun part because the scholars are really the star of the show here. And this has been such an impactful gift from the Steads. I think all of the scholars that have had this support have really found this to be transformational in their careers. And so very much thank you for that gift. So I'm going to just get this going. And the first person that's going to speak is Jeremiah Johnson, who's an associate professor in the Department of Microbiology and Immunology. Jeremiah is an investigator that studies a bacterium called Campylobacter jejuni. It is a foodborne pathogen that causes diarrheal diseases that can be really quite severe. He has been trying to understand how that bacterium invades the gastrointestinal tract and also how it stimulates white cells to put out inflammatory factors that cause a lot of the disease that many humans unfortunately have suffered from. What he's hoping to use the Stead Scholar investment for is really trying to understand whether there's a difference in how the bacterium affects pediatric patients and adults. And I think is looking at opportunities to do some comparing and contrasting to completely understand that pathogenesis. So, Jeremiah?
All right. Thank you, everybody, for showing up today to hear a brief overview of our talk on the project that we had funded by the Stead Family Scholars program, where we're going to be looking at the impact of Campylobacter infection on pediatric intestinal health. And so I like to start by reminding everybody that diarrheal diseases are still a leading cause of global morbidity and mortality. in the world. So there's almost 4.5 billion cases of diarrheal disease in the world every year. And a lot of these are going to be due to various gastrointestinal pathogens, so whether viral or bacterial. This scope of the problem leads to a significant loss of life, so anywhere from 1.2 to almost 2.5 million human fatalities every year due to diarrheal disease. And this makes diarrheal diseases the number 8 leading cause of human mortality in the world. Unfortunately, the burden of these types of infections is not evenly distributed. So low and middle income countries tend to be the most drastically impacted. And a lot of this has to do with the fact that they really don't have adequate wastewater treatment resources. So this is actually a latrine built over a river that children are playing in. So this is how a lot of these communities tend to get rid of their sewage. And then these communities also tend to be closely associated with their livestock. And so both of these are instances where there's a pretty high chance of these organisms being transmitted from into humans and cause these types of infections. And so when we look at really the who's who of cause these types of infections, it's certainly enterotoxigenic E. coli is a really well-known one that I'm sure everybody's familiar with, as well as non-typhoidal salmonella. Um, Again, a lot of infections and a lot of deaths caused by these particular types of organisms. But the organism that we work on in the lab, as Dean Winokur said, is Campylobacter species. So we don't have quite the publicist, it seems, as E. coli and salmonella have. But certainly these organisms do cause a lot of infections and a lot of deaths. And one of the reasons that they cause a lot of infections is that these organisms reside at high numbers within a lot of agriculturally relevant animals, poultry, cattle, and then can be easily transmitted to humans through the consumption of undercooked meat or the consumption of unpasteurized dairy or the consumption of agriculturally contaminated surface water. And in more developed countries like the United States, these tend to be adult infections, where the bacterium gets ingested and colonizes the gastrointestinal tract and causes this really severe inflammatory gastroenteritis that's characterized by stomach cramps and fever and bloody diarrhea. But overseas, what we find is that it tends to be more of a pediatric type of infection. And so when we look at all of those infections and deaths that I was describing earlier, more than half of those infections actually occur within children under 5 years old. And more than half of the deaths occur in children under 5 years old. And so there's been a lot of work by the Gates Foundation and WHO to really try to understand, what is this that's going on within these pediatric populations. They've tried to identify the source of infection, but it tends to be mostly unknown, but they do believe that it's likely contaminated water. And even if the child doesn't succumb to infection, they have found, again, Gates Foundation and WHO, that these children can be persistently infected with Campylobacter. And this persistent infection winds up to causing the development of a really poorly characterized pathology called environmental enteric dysfunction. And what they find is that, you may have seen those videos, trying to, fundraising to get food for, underserved children. And what they found is that regardless of what you feed those kids, they're always really on this reduced trajectory for growth and development. And they believe that it's because of this environmental enteric dysfunction that drives decreased nutrient acquisition, growth, and cognitive development. And Campylobacter is associated with this outcome. And so the big question that we wanted to use the Stead Family Scholars funds for is really addressing the question of why. And so there's obviously a lot of variables at play within these children, but one of the things that we're hypothesizing is that there's likely an immunological component to what's happening in these kids. And so our lab has really pioneered the study of neutrophils and how neutrophils are involved in the gastroenteritis that occurs during sort of normal adult, intestinal disease in the developed world. Neutrophils are the most abundant white blood cells in humans. So about 80%, up to 80% of your white cells will be these types of white cells. And these are the first responders to the site of most infections. So they're really the pawn on the chessboard of the immune system. And um, essentially, the way that they work is that once they're recruited, they undergo these various processes that lead to the release of various molecules that will eventually harm the invading pathogen, reduce the viability of the pathogen, and hopefully promote the clearance of infection. But one of the problems with this is that a lot of those products aren't specifically going to damage bacterial cells. Instead, what they're going to do in some instances is that they can damage some of our own host tissues, as well as promote the development of even autoimmune disorders. So because of this, there's a high association of neutrophils with numerous inflammatory disorders. One that's most relevant to the system that we study, meaning the gastrointestinal tract, is high neutrophil involvement in severity of inflammatory bowel disease like Crohn's and colitis. And one of the things that we found in my group is that these neutrophils, these white cells, are recruited in high numbers during Campylobacter infection, much higher numbers than you would typically see in most gastrointestinal infections. And that the functions of those white cells, if you can ameliorate those functions, you can actually reduce intestinal disease. And so, um, you know, we want to take sort of this expertise that we've developed and pursue this line of questioning that's based on, again, not a ton of data from the pediatric field, but some data that indicates that pediatric neutrophils and pediatric gastrointestinal tracts, not surprisingly, are inherently different than adults. So all of this stuff that we've really been doing with adult types of models, we need to make it more specific towards sort of a pediatric context. And so in order to do this, we're actually taking pediatric neutrophils and understanding how they respond to this bacterium. We're taking umbilical cord blood and examining those responses. We're also going to the eventual development of hopefully treatments. We need some sort of an animal model to examine this. And so we're going to isolate neutrophils from juvenile mice and baby mice to see whether or not their neutrophils exhibit similar responses to human pediatric neutrophils. And then really extending that work to see how do those activated neutrophils specifically impact the unique properties of pediatric intestinal tissue. And so we're going to get intestinal tissue from pediatric patients and then use stimulated neutrophils to essentially see how is the health of that tissue affected in that environment. And again, developing a pediatric mouse model to see whether or not those mice develop some similar intestinal defects that occur within children overseas in the, again, in the development of this environmental enteric dysfunction that I was talking about. So again, we can hopefully develop treatments that can at least reduce, if not eliminate, some of these impacts on those pediatric populations. And so we've been able to make a decent amount of current progress already. So we have been able to isolate viable neutrophils from umbilical cord blood. We've taken those neutrophils and have been able to infect them with Campylobacter. So what you'll see here, these are adult neutrophils isolated on the same day as these two pediatric samples. And essentially what you're looking at is neutrophils will kind of gobble up a bacterium. That's part of one of the processes that they do. And you can see that the adult neutrophils take up more of the bacterium than, for instance, the pediatric neutrophils. So already an indication that pediatric neutrophils are different in their response to Campylobacter. Then we also looked at the ability of those neutrophils to activate in the presence of the bacteria. And you can see that compared to adult neutrophils, where you get this robust activation, we see little to almost no activation of those white cells by this bacterium. One of the other things we found, which was really kind of astonishing to us, was that there's this really highly inflammatory response that neutrophils can undergo. Again, that's associated with a lot of those inflammatory disorders I talked about. And that process is the production of what's called a net. So this is where the neutrophil essentially just spills out its DNA in response to the infection. And about 15% of the adult neutrophils can undergo this process. But what we see is almost 50% of pediatric neutrophils will undergo this process, indicating that those neutrophil responses are likely going to contribute to an environment that's going to be even more deleterious to gastrointestinal tissue than adult cells. And so we're going to be moving forward with this again to really understand the impact of this activation on pediatric gut health. And so with that, I just want to thank my lab. They're here today, obviously. I couldn't do this without them. And then, of course, two collaborators here at Iowa that are very important to this work. Dr. Jennifer Burmick, who previous Stead scholar, who's helping with some of the pediatric work, as well as Dr. Sarah Short, who's going to help with some of the gastrointestinal work. And then certainly, thank you to the Stead Family Scholars for their support of this important work. And so with that, I'd be happy to take any questions.
Questions for Jeremiah?
Since the mortality from these infections tends to follow the U-shaped curve, so also bad in the elder. Have you considered also studying this in neutrophils with aging?
Yeah, so we actually just had a seminar speaker this last week in our department who looks more at aged neutrophils, and we were talking about exactly doing that as well. They're in the developed world, the elderly population tend to have almost like a similar response as pediatric patients in the developing world. So older people actually have trouble clearing the infection similar to those pediatric patients. So it is an interest, but at least for right now, we're focusing on peds.
Do you have a sense of what's the, let's see if I can remember this right,
Sure.
environmental
Enteric dysfunction.
or
Yep.
dysfunction?
Yeah.
What causes that persistent phenotype or deficit layer? Is there change to the epithelium of dysplasia of some sort? Or
Yeah, so the problem with a lot of these communities, obviously, is they really lack the diagnostic, resources to do like histology and things like that. The best that they've gotten is looking at intestinal markers of increased gut permeability. And so that they find that these children exhibit increased permeability just again using like a fecal marker of that. But beyond kind of the more specific cell biological reason for that, it's not really known. And again, part of what we're going to try to get at.
I'm curious, any vaccine give assessment against Camelback?
Yeah, so one of the things I didn't have time to talk about today, obviously, is warfighters are actually really susceptible to Campylobacter infection. It's one of the big, infections that when somebody gets sent overseas with the Army or Marines, they get Campylobacter. It causes travelers diarrhea. And so the DoD has put a lot of money into trying to find vaccines. There's been some large scale studies and none of the vaccines have worked yet. So.
Are farm mice infected?
What's that?
Are farm mice infected?
Farm mice. Oh my gosh. I don't know anybody that's looked at farm mice.
Seeing that they're around the cattle, you expect that, and it might be an interesting system for looking.
Yeah. So when you get in,
Clean mice, right?
yeah, these yeah.
That would be interesting to look at the mice that might actually have the infection.
Yeah, they've looked at people in sort of the food science ag realm have looked at a lot of at least similar but not mice types of opportunities for transmission. The two organisms that they found that are pretty highly associated with transmission on the farms are wild birds. So, crows and things obviously carry it, and flies. So they found flies can carry it in a herd as well. Okay.
Thank you.
All right. Keep that.
I don't think of pediatric mice as an interesting concept. Okay, our next scholar is Linda Sun, who is an associate professor in the Stead Family Department of Pediatrics in the Division of Nephrology. And she, throughout her career, has been focusing on the transport systems in kidney cells, and very specifically on this dynein-powered transport system. Dynein is a motor protein. It is really critical for transporting water and helping set up filtration systems in the kidney, and also transporting proteins. And when that system goes awry, you have problems with kidney filtration of various substances. What's really remarkable is her lab was one of the first to understand that diabetes actually dysregulates this dynein-powered transport system and ultimately can lead to problems with kidney filtration. So really important work, and what she's going to do is continue those studies trying to understand the impact that diabetes has on this dynein transport system, with the hopes eventually of finding compounds that might disrupt or prevent the damage associated with diabetes. She has the luxury of working in pediatrics, where the hope is that you might be able to understand something very early and find that preventive therapy.
Thank you so much for the nice introduction. Today, I'm very happy to share with you our work on the cells highway system. So some of the cells highway are powered by this motor protein called dynein. It may be very new to many of you. So we found in diabetic kidney cells, this cells highway falls into chaos. So with the support of Stead Family funding, we plan to do innovative research to understand how this causes damage to the kidney cells and how we could fix this problem. So diabetic kidney disease, or DKD, is the leading cause of the kidney failure all over the world. In the United States, nearly half of all the kidney failure cases are due to diabetes. Despite all the advances in anti-diabetic treatment, still 30% of patients with type 1 diabetes and 40% with type 2 eventually enter end-stage renal disease. This plot just to show you how the DKD progresses from early stage to late stage over decades of years. It's a very chronic process. Unfortunately, the current and the emergent new treatments are primarily targeting the late stage, which is already likely not reversible anymore. They may slow down the progression of the disease, but they cannot prevent the disease from the root. So the early stage involves children, adolescents, and young adults. This is a potentially reversible process. Also, unfortunately, there's non-existing treatment. So as a pediatric nephrologist, we have these missions of understand the earliest mechanism, develop new treatment to manage the problem at the early stage, and then eventually protect the kidney before it gets too late. We all know that the diabetes hurts the kidney, but the big question is how. Our research found this problem starts with these little blue cells called podocytes. In healthy condition, they form the filtering units of the kidney. And in diabetes, these filtering units fall apart, and the protein will leak into the urine and eventually cause kidney scars. At this time, the primary mechanism behind this phenomenon is largely unknown. So when our outstanding endocrinologist is trying to develop new treatment to treat diabetes, what can we do to protect the podocyte? So here is how our story comes in. We study the transportation system inside the podocytes. Just imagine podocytes are like small cities. In order to make this city functioning, important cargoes will need to be delivered to the correct place at the correct time. So here we study one of the important cargoes called nephrin. It's this red labeled protein. Let me show you a live cell imaging. Here you can see the green labeled dynein sends the red label nephrin to the correct place at the right time to maintain the proper function of the trafficking system, proper function of the kidney filtration system. In healthy podocytes, dynein mediates the transitional balance of nephrin between the recycling system and the retrenching system. This will allow adequate nephrin to be delivered to the filtering unit to maintain the integrity of the kidney function. However, in diabetes, the dynein-mediated trafficking system gets dysregulated and it ends up sending too much nephrin to the trenching system. As a result, the filtering units will disappear. Our research found this mistrafficking event in diabetes actually is resulted from the overexpression of a list of dynein genes. Here we can see those red-colored upregulation. So we call this phenomena of overexpression upregulation. More importantly, this upregulation was found to correlate with the severity of the disease and decline of renal function. This underscores how important this new mechanism could be. So next question we try to answer is, can we protect the podocyte by correcting dynein under diabetic condition? So in animal models, we tried two strategies. Firstly, we use a genetic tool to turn off dynein in diabetic kidney cells. And then in the second strategy, because we found diabetes upregulates all the dynein genes through a gene commander called SP1. So we use a chemical compound to block SP1 and in order to turn down dynein. In those animal models, we find both of the strategy was able to restore the proper trafficking and protect the podocytes. So those two strategies I talked about are conceptual. They're not ready for clinical use yet. So with the support of Stead Family's funding, we plan to take a high-risk, high-rewarding translational path. So we developed this high-throughput drug screening platform. We use that to screen FDA-approved drug libraries with the goal of identifying and repurposing existing drugs. Those drugs has already been tested in human beings for the safety, and those candidates will have a better chance of safely normalize the dynein in patients and be more ready for clinical use. So with the Stead Family support, we also plan to take the dynein discovery to the next level. Our recent research identified a new role of dynein beyond its traditional role in trafficking. So specifically, we find a connection between dynein and organelles, gene regulators important for cell metabolism. More interestingly, we find also there seems to be a connection between dynein and the gene regulators responsive to sex hormones. So these findings just lead to two new directions. So first, through the interactions between dynein and these organelles, gene regulators for metabolism, we try to understand the new role of dynein in reshaping the cell metabolism and reprogramming gene transcription leading to the kidney injury. So this piece of new knowledge will help us to design new treatment that could restore the cell metabolism. So these are the strategies we proposed. The direction 2. Through the connections of dynein with the specific gene regulators responsive to sex hormones, we try to test this new idea, a new hypothesis that dynein may drive a sex-specific mechanism for diabetic kidney disease. So this new knowledge eventually will impact our understanding of the different disease susceptibility and the way we treat the diabetic kidney disease in boys versus girls, in male versus female. So in summary, to wrap everything up, early stage diabetic kidney disease is often overlooked in young adults, pediatric patients, and adolescents. But it actually gives us the opportunity to investigate and intervene. The research we try to pursue is we try to understand during this golden window how dynein misregulation disrupts the trafficking, rewires the metabolism, and scrambles the gene transcription. And hopefully the new findings will lead us to new treatment that could restore all these balances. And so we can protect the kidney before it gets before the damages become irreversible. Hopefully, by doing all this, the pediatric nephrologist can protect the children's kidney today and provide them a better future tomorrow. Last, I would like to express my sincere gratitude to my research team. They are at the back. And then my collaborators and my funding agencies, specifically the Stead Family Funding, for supporting our daring, high-risk, high-rewarding approaches leading to discovery. Thank you for all your attention. I'm ready to take questions. Yes, please.
Very interesting. For your high throughput drug screen, I wonder what type of the cell or system you're gonna use. Is that the podocyte, please?
That's a good question. So, in order to make the high throughput screening cells viable, we decided to use U2OS cell, which is the osteosarcoma cell line. And we developed a cell line that could co-expressing two big fusion proteins. Once we give rapamycin treatment, they can form the rapa-induced cross-link, so we can track the dyne-mediated trafficking of certain cargo. So in order to make the cells really viable and go through all the screenings, we decided to use this U2OS cell, which has better viability. But eventually, for verification, we have to do all the studies in podocytes.
Related, I think you mentioned this is a FDA approved Belagistan to repurpose, and I wonder how big is your library and
So,
target that?
yeah, good question. So for the first pilot screening, we plan to do Presswick drug library, which only have like 1,700 FDA drugs and for repurposing. And then the primary screening will be, we plan to use the broad Harvard Institute repurposing library that include close to 7,000 drugs. But beyond that, we'll see if we can have any positive hit of either dyne activator or inhibitor. And beyond that, I have to think about, because based on my goal, I try to find those safer candidates. But also at the same time, if we can find any candidate that's suggesting any new functions, we may be able to expand it to certain category of drug chemical compound to expand the library.
Thank you.
You do suggest that dying is playing a role in gene regulation. Is there a pool of the dyne that's localized in the nucleus, or do you think these are all kind of indirect effects to the cytoplasm? Both factor receptors and signal transduction pathways,
So dyne, because the cell dividing, the nuclear dividing doesn't involve a very active dyne function. So I think one of the primary function does include its role in recruiting important proteins to mediate the meiosis process. That's one of the function I haven't studied yet, but I think Jillian, my graduate student, have a big interest in that. That will be something we plan to look into that. I also connect our research to aging and cancer, but it's a little bit sideway. Our current primary focus is on the in the stable mature cell differentiated what it functions. But eventually, I think that's actually a pretty good direction and may play a very important role.
Can you elaborate on sex hormone connection? Is there a difference between male-activated or susceptibility?
Yeah, so
May please.
so here I didn't have the space to include one of the slide. They did show in diabetic kidney disease. They monitored the disease progression in males and females. Obviously, at the pre-middle age group, females has much lower development and progression of diabetic kidney disease compared to male. Then, in our study, here I didn't include the gene regulators here. So we did spatial transcriptomics and tried to find dyne-dependent proteins or genes potentially involved in the development of diabetic podocyte injury. And we actually did enrichment. We find some regulators responsive to estrogen. One of them is like estrogen receptor 1. It's very prominent. There are a list of genes, either upregulated or downregulated, that could be enriched into this transcriptional factor. Also, there are estrogen receptor-related proteins like ESRAA. So that's estrogen-related. But also we find the connection between dynein and the progesterone receptor pathway. Also, there are sometimes involved some transcriptional factors involved in the androgen pathway. When I look into the downstream, they are primarily SOX1, SOX2, not SOX9. So that's some type of sex hormone responsive gene regulators, not necessarily directly related to like sex development, but could be involved in cell differentiation, fibrosis, inflammation, oxidative stresses. So those are potentially tell me some mechanism that may drive a different sex susceptibility.
Then I have a different kind of question for you. So you were at Harvard doing great. You were about to get an NIH award, and you came here. You gave up that award to come to the University of Iowa. You did great here. You went straight from our NIH Career Development Award that we have at Iowa to an RO1, which is very unusual. And you're staying here. So what made you come here, and what keeps you here?
So I have to, firstly, I have to thank Lindsay Hushman. At the critical time point when I was trying to be independent, I met Lindsay Hushman at ASA conference, and I just find all these young faculties compared to the peer faculties in Harvard. She just did so well, and everybody like junior peers in Iowa, they were able to have their own career. So that's definitely just evidence-based. Then when I moved here, I just appreciate so much space that I got, and also the support from my department, my division, and the graduate school program. They allow me to work with graduate students maybe at the early stage. So those are like a really rich experience for me to grow. And I am self-evident that's the correct choice.
So I like the concept of the high risk, high rewards. And I think that's what this funding allows our investigators to pursue, is some of that work that's really a little bit more early exploration that isn't yet ready for grant funding. And I think this is a really important point of this funding. Last but not least, we have Dr. Stephanie Gantz, who's going to speak. She's an assistant professor in the Department of Molecular Physiology and Biophysics. She studies a brain receptor, delta-1 glutamate receptors, which are really important for the developing synapses in the brain. These, when they become dysfunctional, have been associated with a variety of different neuropsychiatric diseases, from schizophrenia to bipolar disorder, even autism spectrum disorder. She has been really involved in understanding the fundamental science of these delta glutamate receptors. And what she's hoping to do with the Stead Family funding is explore pH and its effect on these delta glutamate receptors, and kind of correlate that with some of the symptoms that you see, cognition, and some of the symptoms that are associated with some of these neuropsychiatric disorders. We like receptors in the brain because we've found that we can find drug compounds for some receptors in the brain. And so that's the ultimate goal, is to find compounds that can be used to change the modulation of these delta glutamate receptors. So Stephanie?
Thank you so much for that introduction. That was basically what I have to say. I'm really delighted to be here to share some of what's going on in our lab. But before I start into the science, I just want to take a moment to express our gratitude to the Stead family for your exceptional generosity to the University of Iowa and to the support of our research program. And I want to take a moment to introduce you to some of the team that are working on these projects. They're also in the back. That's the lab area back there. I have 2 graduate students that are native Iowans that are working hard on these projects. I also have 2 postbac research interns that we recruited to move here to work on our science. So my lab studies the brain. And when you think about the brain, sometimes we use the analogy of it being like a computer, and that's not altogether incorrect just because the brain is electric and it's made up of circuits. When we think about the brain, we often think about it encoding in kind of binary, like ones or zeros, go or no go. And that's true, but what's unique about the brain is that it signals electrically on very different time scales. So that go, no go signal can happen really quickly, or it can happen really very slowly. And that diversity in time scale is what allows our brain to do all of the diverse functions it does, right? From detecting sound or light very quickly, or maybe solving a puzzle that might take just a few more moments, all the way down to our sleep-wake cycle, which is a lot longer, and then our mood and affect, which can persist for months and even years for some people. So, the longest lasting electrical signals are known to modulate cognition, which is the way we think and reason about the world, as well as our emotions. And when I think about long lasting electrical signals, I often think about the ocean tide. And in this project, a photographer was taking an image at the same location at low and high tide to demonstrate the remarkable differences in the landscape. And this long-lasting electrical signal changes the way that discrete signals are interpreted. So if our ocean tide is our long-lasting signal or resting membrane potential, then individual waves, or these crests, might be discrete signals that are coming in. So in this cartoon version on the bottom here, I'm showing high tide and low tide. And let's just imagine for a moment that the brain has to reach this gold bar in order to signal output. Then you can see that that would happen in high tide and it wouldn't happen in low tide. And the brain works this way electrically, but I think that we've all experienced the physiological relevance or outcome of this. Anger is a great example. If something irritates you, then all of a sudden, all of these other little things that might not have otherwise triggered you just really grind at you. The contrary is true as well. So in terms of cognition, way we think and reason, if you're really sleepy or you're mentally fatigued, you might have a lot of signals coming in and you just really can't put the pieces together. Now, I'm using this as an example of transient things, but for many individuals, mood and affect or cognition issues persist for a long time. So when we ask the question, how can we better treat a decline in cognition or a decline in mental health, I often think, what can change the tide? There's going to be instances where we want it to be lower, and there's instances where we want it to be higher. So we started thinking, what can change the tide? And there's this finding in the literature that there's a decrease in brain pH that's associated with cognitive decline and neuropsychiatric symptoms. The brain can experience a deviation in pH from a whole number of disorders or acute issues like stroke or heart attack, but also from lung disease, pneumonia, sleep apnea, as well as pain, stress, and anxiety-related hyperventilation. So the unresolved questions is, how does the brain regulate pH? And what in the brain senses a shift in pH to create a really long-lasting change in neural activity? And now, my lab is pursuing a totally unexplored and novel target, this delta-1 glutamate receptor, which we'll try and make a case is a promising target. So you might be asking yourself, well, what on earth is delta-1 glutamate receptor? So the delta-1s, which we call GLUD1 for short, are a member of the ionotropic glutamate receptor family. When I say receptor, this is a protein in the brain that senses or receives a certain chemical, and then it can open a pore in the membrane, and that pore or channel allows charged particles to flow through, and that charge is what carries electrical currents. We know that GLUD generates a really long-lasting excitatory current. And when I say excitatory, that's kind of like the green light. That's the go signal for the brain where these receptors are expressed. And they're also found in nearly every brain region in early development into adulthood. This is where it gets really interesting. Despite its name as a glutamate receptor, it does not bind nor does it open to glutamate. So in this cartoon, I'm showing you what a receptor might look like. This is that pore where it's closed, and here an agonist binds. The pore opens, and then sodium carrying its positive charge flows in, and that's what's generating electrical currents. So when you're using the term agonist, it's kind of like cash or easy pass at the toll booth. You have to have it to open the gate. So not only does it not open to glutamate, but for the longest time, there was no way to open GLUD1. This has really been a critical barrier towards advancing our understanding of these receptors. It's kind of like we're stuck at the gate and we don't have our easy pass. We can't learn anything about what's on the other side if we can't open that gate. And I'm using this graph here to illustrate exactly how critical of a barrier this has been. So this is the number of publications versus year for the NMDA receptor, which is another glutamate receptor, and that's shown in gray. And this isn't a mistake. Down here, you can hardly see it. The gold, that's for GLUD. And so a couple features I'd like you to notice here. One, GLUD is incredibly understudied. Two, something really interesting happened in the early 80s, right here, right? Because it used to be right along with GLUD. What happened then? That's when they figured out how to open the gate. So it's pivotal because as soon as they learned how to do that, our understanding just skyrocketed. So I think that we are right at that inflection point, which is a moment that I want to highlight, that this is why these funds coming are such a pivotal moment, because I think we're on the forefront of history. My lab made a significant breakthrough. We found out how to open the gates. And now it's kind of interesting. So we found this compound noradrenaline. This is an endogenous chemical in your body, and it opens GLUD1. All of our work is done in acute mouse brain slices. And so you're seeing an image. This is what the brain slice looks like underneath our microscope. Here is a brain cell. And then this is a little glass pipette. That's how we record electrical current. So I've just taken this image and made a cartoon diagram here. We found that noradrenaline is actually binding to another receptor, this alpha-1 adrenergic receptor. And then ways that we don't understand yet is talking to GLUD1 to open the gate. So at least we have a tool now that we can open it. And we found that GLUD1 carries this really long-lasting excitatory current. So when we come back to our tide analogy, when it's open, it's like high tide and it's sending a strong go signal. So now let's revisit the idea of what's happening when we decrease brain pH. And a graduate student in my lab, Drew Kane, tested this, and he found that by shifting from neutral pH to slightly acidic at 6.8, there's a profound reduction in the magnitude of that current. That is we predict that that's like taking a strong green light for those neurons and shifting it to a yellow light. Things are slowing down. So with this award, we'd really like to examine the relationship between the alpha-1 adrenergic receptor, GLUD1 receptors, and brain pH. We know that when there is a decrease in brain pH, there's a decline in cognition and in mental health. We also know that there's a decrease in this GLUD1 currents, and so the question that we're asking is whether it's the decrease in GLUD1 current that's contributing to the decline in cognition and mental health. If everything goes to plan, we might be looking at GLUD1 as a novel pharmaceutical target for cognitive decline and neuropsychiatric symptoms. Currently, there are no pharmaceuticals available to target GLUD1 selectively. And so we imagine that our research will provide the necessary impetus for the development of GLUD1 targeted pharmacotherapies. So with that, Again, thank you so much for your attention. I'm happy to answer questions.
Non-scientific question, but how do you detect? What do you use to detect this in patients with the pH and all that? How do you diagnose? If that makes sense.
How do you diagnose the change in pH?
Yeah.
So that's outside of what my lab does. We're building on a foundation. There are actually excellent clinicians here that do it. There are postmortem is one that we originally used, but there's also abilities to image it using MRI. So you can actually detect changes in pH, and we can do a little bit of that in rodent models as well. And in rodent models, you can have them breathe higher levels of CO2, and you can see that decrease in brain pH. Yes.
Maybe I missed. I'm curious. D1's expression in the brain, is that for all type of a cell or particular type of cell you want?
Yeah, so it's found in almost nearly every brain region, but there is cell type specificity within those regions. So there has to be some patterning that's important. A lot of people have looked at that only in rodents, to my knowledge, but it is pretty much everywhere.
I'm also curious, is aging cause the decline the activity or expression of your gene?
What was
Aging?
aging? Oh, great question. Totally unexplored. So all of the changes with GLUD over lifetime, completely unexplored. Also, whether it changes with experience, or something in my lab is really interesting is exposure to alcohol or drugs of abuse. All of that has yet to be done. Yeah. Yes.
Is the pH, do you guys think affecting just the booty part or both parts of the system there with Norco?
Yeah, that's a really great question. There's a lot of work that has to be done to determine that, right? Because changing pH will likely change a lot of things in the brain. We have reason to believe that it's affecting the channel directly, and that is the primary underlying cause of it being so reduced. It doesn't mean that there aren't other things happening, but it's probably affecting the channel directly. Yes.
So extracellular pH homeostasis is not completely well understood, especially when it comes to the sensing portion, which is of what you're studying. Is very interesting. Have you looked at co-express PD-1 with adrenoceptors in other organs where you could kind of like extend your research to? And in some cases, it may be easier to find models to study in vivo.
Right. I think that's a fantastic question. One of the limitations right now is imaging GLUD1 in other tissues. The antibodies have largely failed strategies. So what it would necessitate is probably putting a fluorescent tag on it, and it's definitely something that we're interested in. In other tissues, I think is a great idea. It does show up elsewhere in the brain where we don't know what it does. Yes.
His question. Do you think that because of pH change, autobahn receptors send some intercellular signals and then they can cause some glutamate D1 expression in the memory? Because as far as I know, receptor expression is kind of dynamic process, and GLUD1s are not always there, right? They are just appear in membrane according to communicate change, or are they appearing in the memory coupled to autobahn receptors intercellular signals?
Right. So the question, if I'm understanding correctly, is are we looking at trafficking of GLUD1 coming to the membrane with either protons or with alpha-1? We don't know. That's something that we're pursuing, and we have reason to believe that in part that might be what's happening, is that they're moving in and out. Still, yeah, I feel like all of this is still open questions. Yeah, but it's a great one that we're pursuing. Okay, thank you.
Wow, those were terrific. And I'm going to remember the low tide, high tide for years now. So thank you for explaining it in a way that I could understand. So, and thanks to all of you for being here. A special shout out to the students. Can you just raise your hand if you're a student? Thanks all for being here. And we hope that you'll stay here for many future parts of your career here in Iowa. So thanks for being here, and thanks a special thanks to your contributions. You work behind the scenes to make a lot of this possible. So I love the fact that the scholars thanked them all. Special thank you, of course, to Jerry, Tiffany, and Dana for being here, and looking forward to the continued success of the Stead Innovation Scholars Program with its new name. So thanks so much.